morning, good afternoon to everyone in the United States, and especially to our Dr. Carla, who is calling in from Spain, Barcelona to be precise. Today we are having this very important webinar for the benefit of everyone who's interested in the field of Alzheimer's, dementia, and Lewy bodies. The event is supported by Brain Support Network, as well as Stanford University. My affiliation with BSN is I'm on the board. I also am the daughter of an LBD patient who was diagnosed in 2007 at UC Davis and UCSF. And my father had been exhibiting symptoms many years before, but we didn't know what was going on. So it's important to identify the symptoms early on so you can get the right treatment, medication, physical therapy, and so forth. The guest speaker today is a wonderful collaborator with BSN, Carla Abdelnour, originally from Venezuela, now living in Spain, as I mentioned. And she has been at Stanford working on her postdoctoral research focusing on Lewy body dementia. But currently she is certainly an MD who specializes in movement disorder and dementia. And it is a specialty of hers, which we are appreciative that we have someone with her knowledge and experience and passion for the topic with us today. So I'd like to invite Carla to say a few words and then we can get started.
Great. Thank you very much, Linda. I really appreciate the opportunity to do this presentation today, and I'm very grateful for your introduction. So now I'm going to share my screen, and please let me know if you see my slides. Let me turn that to presentation mode and continue to share. Do you see the presentation mode or?
Yes.
Okay, perfect. But the presentation mode or the first slide only, because I never know. I have to.
Oh, the first slide.
The first slide only. Okay, perfect. Sometimes I don't know which slide it is. Okay, great. So first I want to start today because I'm a neurologist about talking about cognitive function. So there are different cognitive functions, and if I were to ask you or ask anybody what is their favorite one, maybe some people will say that they like their memory or language. I think the brain is an amazing organ. I'm obsessed about the brain. But I want to share with you that actually my favorite function is social cognition. So we as humans have developed a special part in the brain where we have functions to collaborate in communities. So these functions are, we have the ability to think that other people are what other people might be thinking. We're able to identify people's emotions, and we're also able to show empathy. So put ourselves in the shoes of others. And those are functions, oh, so sorry. And those are functions that actually are in a specific part of the brain. And thanks to those functions is that we are able to collaborate and to solve very complex problems. So I think as we move along during the presentation today, I want to make clear that there are a lot of things that we are working on and future directions, but we're working in these together as a team. So today I'm going to talk about what are the new things that, or the future directions, or new things that we're working on in Lewy body dementia. But first, I want to start by defining what is Lewy body dementia. So Lewy body dementia is a neurodegenerative disorder, meaning that the neurons in the brain are suffering from this condition and they are dying. And when these neurons die, the neurons that are alive take on the functions of the neurons that are dead. But it comes to a point with the neurons that are alive can't take up those many tasks. So we're starting to see deficits. And in particular, in Lewy body dementia, we have a combination of symptoms that are characterized by movement disorders and cognitive disorders. So it's a very special type of neurodegenerative disorder. So people that have normal cognition, in this case, will develop mild cognitive impairment and at the end, dementia. And the difference between having mild cognitive impairment of dementia is if the person has the ability to live independently. So if you're able to do activities of daily living by yourself. So a person that has mild cognitive impairment due to a neurocognitive disorder is independent for activities of daily living, although that person have cognitive deficits. But a person with dementia, the word dementia, what it means is that you need help from another person to perform activities of daily living, even if those tasks are not basic tasks like eating or bathing. At the beginning, those are maintained, but where are the ones that are affected are the instrumental activities of daily living. So your ability to manage finances or take your medication or manage your calendar, for example. So Lewy body dementia is an umbrella term that includes both Parkinson's disease and dementia with Lewy bodies. And so a person that is diagnosed with Parkinson's disease may or may not present with cognitive impairment. And then I'm going to talk about a little bit about the symptoms that people with Parkinson's disease and what is the difference with dementia with Lewy bodies. But let's say a person with Parkinson's disease presents without cognitive impairment. So this person will present a slowness in movement or tremor with or without tremor or rigidity. And then in a percentage of these people will develop mild cognitive impairment and eventually will develop dementia. So there are studies that have shown that if we wait enough time, people that without cognitive impairment that have Parkinson's disease will eventually develop dementia. But in the case of dementia with Lewy bodies, it seems like that process is more rapid, it's accelerated. So usually they present the cognitive and the motor symptoms in a short period of time. So if we ask ourselves then, what is the difference between Parkinson's disease and dementia with Lewy bodies, is actually time. And the way that we clinicians make this differentiation is by setting up a rule that is called the 1-year rule. So if a person presents within 1 year with cognitive and motor symptoms of Parkinson's disease, we call that dementia with Lewy bodies. But if the person presents first with motor symptoms and after more than 1 year that person develops cognitive symptoms, we call that Parkinson's disease. So there are a lot of similarities between these diseases, but this is the way that we classify them in the clinic. But if you want to know more about what is dementia with Lewy bodies or Lewy body dementia, I encourage you to look for this video that has been shared online in YouTube. So if you write on YouTube Sue's stories, you will find this video, and Sue is an amazing person. And the Susan and Charles Berhoff Foundation prepared this video, and she explains her experience living with Lewy body dementia. Now let's move on. What is new in the field of Lewy body dementia? So actually, there are a lot of things, and I'm not going to be able to cover all the studies that are being done in the field. So I summarize them. There are clinical studies. There are studies on underrepresented populations. There are studies focused on differences in sex and gender. And there are also studies on biomarkers and treatments. The studies that I'm going to focus on today are the studies on biomarkers and treatment, because I'm more focused on those, but the other ones are very relevant also, and this is a very active field. So the part one, let's talk about biomarkers. So what is a biomarker? When I say that, what do I mean? And I copy here the definition from the Federal Drug Administration. And what they say is that a biomarker is a characteristic that we can measure, and is an indicator of a normal biological process. So if we're healthy, or a pathological process, meaning if we have a disease. It can also be a measure of the response of a treatment. So if I take a blood sample and I'm able to calculate something in blood that tells me that that person has responded to a treatment, that is also a biomarker. So that means that this characteristic could be molecular, histologic, meaning that tissues, or images from radiographic, or also can be physiological characteristics. All of these are types of biomarker. Now, what is not a biomarker is the assessment of how an individual feels, functions, or survives. So, the Federal Drug Administration, the FDA, have further categorized the biomarkers, and I'm going to explain some of these categories. First, we have diagnostic biomarkers, and those are biomarkers or measures that we take to diagnose a disease. So if I take a blood sample and I measure, for example, the glucose levels, I can diagnose a person with diabetes. So that will be a diagnostic biomarker. Then there are prognostic biomarkers, and these biomarkers are the ones that tell me what is going to be the future of that person with that disease. So is this person going to have a more rapid progression or not? How is going to be the course of the disease? And that is a prognostic biomarker. So for example, if I obtain a measure in blood, and I have high levels of biomarker X, I can say, well, I know that the studies have shown that people that have high levels of these biomarkers progress more rapid than people that don't. So that will be an example of a prognostic biomarker. Now, a monitoring biomarker is a biomarker that I measure repeatedly. So I take a measure, for example, visit 1, and then I see the patient in 6 months, and I take the same measure, and then I take another measure in 6 months and another 6 months, and so on. And with that information, I'm able to see what is the evolution of the biomarker over time. And that is going to tell me, for example, if the person has a rapid course of the disease or not, or if I give the person a treatment, a monitoring biomarker that changes can tell me if that treatment is working or not. But the case of this type of biomarkers is that you need to measure them repeatedly. And those biomarkers are very important because they'll tell us how the disease is progressing. And then we have predictive biomarkers, and they're biomarkers that we measure at baseline, and it's going to tell us what is the risk of the person of having a particular outcome. So in this case, is this person going to have a shorter survival or not? Is this person going to have a higher mortality rate or not, depending on their biomarker? But that is a person that already has the disease, and I measure another biological characteristic that tells me about the risk. Now, there are other biomarkers that I'm not going to define today. Those are susceptibility risks, there are pharmacodynamic and response and safety. But those have to do more with people that don't have the disease or the people that are in clinical trials. What I want to focus on is what we are doing in biomarker research. Why do we want to look for biomarkers? So what we want to identify these markers because we want to have objective measures of what might be happening in the body, in the biology of the person. So with that, we will improve our ability to diagnose the patients better, because the way that we diagnose patients currently is with clinical symptoms. And those symptoms between different neurodegenerative diseases overlap a lot. So people with Alzheimer's disease have similar symptoms that people with Lewy body dementia, and people with Parkinson disease have very similar symptoms with people that have Lewy body dementia. And doing the clinical differentiation is not always easy. So if we're able to identify what are the biological characteristics of these different diseases, we are going to improve our ability to diagnose people. And we also want to be able to diagnose people early. So currently we're diagnosing people in the dementia stage, but ideally we should be able to diagnose people in the mild cognitive impairment stage, or even early before they develop symptoms. That would be ideal. So I was talking today with Robin, Shepard, and Chuck also, and Robin was telling me that, and Chuck was telling me that he went to the doctor and he received his annual checkup. So I imagine in the future, a person like me or Chuck, you go to the doctor, you do a workup, a blood workup, and you see what are the biomarkers there. I'll tell you if you have increased risk of disease X or disease Y, and including this type of diseases. And that's what we want to see or have in the future. So what we're working on is trying to improve the biomarkers that we already have, but we also need to develop these novel biomarkers to identify people before they have disease. That will be amazing. And for that, we are focusing on biomarkers in biofluids, meaning in blood or in a liquid that is in our nervous system that is called cerebrospinal fluid, or neuroimaging biomarker. So one of the holy grails in Lewy body dementia will be to have a biomarker for alpha-synuclein. I'm going to explain why. So Lewy body dementia is characterized by the abnormal deposition of a protein that we all have in the brain that is called alpha-synuclein. The thing is that in people with Lewy body dementia, this protein starts to clump together. It misfolds, so the protein has a shape, but for some reason that we don't understand, that shape is lost, and that does that these proteins start to clump together and create aggregates inside of the cells of the brain, so inside the neuron. And over time, it seems like having these aggregates of this protein inside the neurons is not good for the neurons, and neurons end up dying. So this is a characteristic that we see in cases diagnosed with Lewy body dementia, particularly. So it would be nice to identify this protein somehow in a biofluid or in neuroimaging, because the issue that we have with neurodegenerative diseases is that we can take a biopsy of the brain and look at that in the microscope. Like I can take a biopsy of my patients, unfortunately, and see that, right? Because we're not going to do that for diagnosis. So we need indirect measures that we can take for a fluid, like the cerebrospinal fluid. This is the fluid that surrounds the brain and the spinal cord. Or if we can take in the blood or urine, it could be saliva, it could be any biofluid, but also a neuroimaging. Let's say I take images of your brain and I'm able to see this protein or other proteins or processes. So those will be indirect measures of what might be happening. And in the case of neuroimaging biomarkers, one of the biomarkers that we have currently is a biomarker that measures the amount of neurons that produce dopamine. So what happens in Lewy body dementia is that the neurons that are mostly affected, or one of the ones that we think are the first affected, are neurons that produce a substance called dopamine, we need dopamine in the brain for a lot of functions, but one of those functions is movement. So that's why people with Parkinson's disease and Lewy body dementia develop movement disorders. So we have the ability to see if the dopamine system is affected in the brain or not with this type of neuroimages, where we take a substance and we inject that in the blood, and that substance goes to the brain and light up different parts of the brain, and that helps us to analyze if the dopamine neurons are dead or not. But currently what we are working on is on quantitative methods to analyze that data, because the majority of those images are analyzed visually, but we need to have more objective measures so we can use those measures over time and potentially use that, for example, as a monitoring biomarker. Also, it will be great to have a tracer or a way to see the amount of alpha-synuclein in the brain. So imagine that I inject you with a radio tracer. That's a substance that will go directly with where the neurons that have alpha-synuclein, or the areas of the brains where there is more alpha-synuclein, and then I take pictures of your brain and those areas light up differently. So we are working on that. And there has been one radio tracer that has been recently published that is able to actually detect alpha-synuclein in the brain. But the type of alpha-synuclein that we're able to detect is the one that is altered in a type of disease that is called multiple system atrophy. So currently, we unfortunately don't have a tracer that detects the alpha-synuclein that aggregates in Parkinson's disease or dementia with Lewy bodies. We're working on that, but it's very difficult to develop this type of tracer because the actual amount of Lewy bodies in the brain is very small. So detecting a signal at the level of the neurons is very difficult. But we're working on it, and I'm hopeful that in the upcoming years we will have a radio tracer. It will be very important for us to see what is the anatomical distribution of this protein in the brain, and also follow up how this protein moves around the brain and from area B to from area A to area B and area C. How does that spread occurs over time? And also, if we develop a treatment that, for example, targets alpha-synuclein, we can use this type of radio tracer to see if the target engagement, how is it called? So if we actually that drug targets that protein or not. What we do have available right now in Parkinson's disease and dementia with Lewy bodies, but it's also there are ways to analyze it for MSA, is a biomarker for alpha-synuclein in the cerebrospinal fluid. So there is a technique called lumbar puncture, and this is a technique that we do as a standard technique for diagnosis in neurology. For example, if I suspect that my patient have a meningitis, I will do a lumbar puncture. Or if I suspect that a person sometimes have for multiple sclerosis, if I want to analyze more information about that type of disease, I can do a lumbar puncture. So the lumbar puncture is a standard procedure, but it's true that involves a needle. And a lot of people don't like needles, which is understandable. So what we will do is put that needle in the spine in a space where there is no spinal cord. There is only nerves or the roots that goes to the legs. And in that space, there is this fluid that surrounds the spinal cord and the brain. So we take a sample of that and we analyze it. And that gave us a lot of information. The good thing about the cerebrospinal fluid is that we can analyze a lot of proteins, a lot of substances at the same time. So there has been a technique developed that is able to detect if that sample have aggregates of alpha-synuclein or not. And it has shown that the number of false positive or false negative cases is very small in the case of Parkinson's disease, and also in cases with dementia with Lewy bodies. So it seems like this is a promising technique, but it's currently this technique is more frequently done in research, although there are some places where it's being done now also in the clinic. I'm not going to spend too much time explaining how it works because it's very technical, and those details, if you have questions, I'm happy to explain it. But basically what happens is that we take a sample of the cerebrospinal fluid and we put a protein that wakes up the proteins that might be dormant that are associated with the alpha-synuclein aggregates after checking the sample, and that reveals a signal that we can detect with a tag, with a label that we see with fluorescence in a machine. And that will tell us if the person have or doesn't have detected alpha-synuclein aggregates in the sample. It doesn't tell us the amount, so we don't get a numerical result. We only get a binary result, like positive or negative, or in this case will be detected or not detected. But another thing that we can do in biofluid, as I was telling you, is analyze more proteins. So now I'm going to move along the relationship. I'm going to explain a little bit more about the relationship between Lewy body disease and other diseases. And I'm using the term Lewy body disease to include people with Parkinson's disease that don't have cognitive impairment. So Alzheimer's disease is characterized by the deposition of amyloid beta in plaques and neurofibrillary tangles. There's a protein that is called tau. So imagine Alzheimer's disease is two proteins, one that we call amyloid and another one that we call tau. Whereas Lewy body disease, we have been talking about, that is characterized by the deposition of this protein that is called alpha-synuclein. However, a lot of cases that we see have mixed pathology. So usually people don't have just one disease, but sometimes they can have two diseases. And I can say that probably one of the first studies that have analyzed this mixed pathology were actually cases of Lewy body dementia or Lewy body disease that use biomarkers for detecting amyloid or tau, but particularly amyloid in people with Lewy body disease. So this is how we started to study this in vivo. We also know this, of course, from autopsy studies because when we analyze the brain of people that have died, we can see the different proteins, and a lot of these brains that have multiple proteins, not just one. So pure cases with only alpha-synuclein deposits are rare. Most cases we have one or two or three more proteinopathies, and there are different combinations, but a frequent combination is having alpha-synuclein with amyloid and or tau. So actually, pure cases are mostly from 10 to 20%, but cases with Lewy body disease and Alzheimer's disease around 50% in biomarker studies, and with Lewy or vascular disease are approximately 30%. So it's very frequent. But why do we want to study this? Is it just because it's frequent? Well, actually, no. The things that these mixed cases are interesting because they have increased risk of nursing home admission, they have shorter survival, a more severe disease course, worse performance on cognitive tests, different clinical features, and they also have different responses to treatment. So we want to be able to identify these cases in the clinic for sure, because what it seems is like they have worse progression overall. Now, we have said that we have biomarkers to identify amyloid and tau, And now we have biomarkers to identify alpha-synuclein. So in the cerebrospinal fluid, we could be able to combine all of these biomarkers to identify these mixed cases. But we also now have another biomarkers that are more novel that have been developed in plasma. And those biomarkers are mainly associated with Alzheimer's disease pathology. So we're able to identify in plasma amyloid beta, phosphorylated tau. We're also able to identify a protein that is called glial fibrillary acidic protein that has to do with inflammation in the brain. And we're able to identify another protein that is called neurofilament light. And this protein is associated with loss of axons, so loss of neurons. It's a biomarker for neurodegeneration and is often high when there are high levels of neurodegeneration. What we don't have yet, but hopefully we'll have in the future, in the upcoming years, is a biomarker in blood for alpha-synuclein, but we're working on it. So the take-home message for this part is that we, in the field of neurodegenerative diseases, have been classifying these diseases in different buckets depending on the symptoms. And from those symptoms, we have identified different proteins. So we say if a person has Alzheimer's disease, we know that they have amyloid and tau, or if they have alpha-synuclein, they have PD or DLB. But the reality is a little bit more complex. And we know now that most cases are actually mixed. And we want to use biomarkers to understand those interactions, to be able to identify people that have mixed pathologies, because we know that they have worse prognosis. So this is what we're currently are, trying to understand these mixed proteinopathies biologically with the use of biomarkers. Now I'm going to move to explaining where are we right now in terms of treatments. I think we have explained, and I think it's important to emphasize that one of the main goals is to be able to identify patients and diagnose them better in the clinic, but also earlier. But the other probably important topic is then treat them, right? We want to have good treatments for our patients, not only for symptoms, but also treatments that decrease the progression of the disease, that target the progression of the disease, or eventually cure the disease. That would be ideal. Before I start explaining what we are in treatments, I want to explain some concepts about how clinical trials work, because the way that we develop treatments is in clinical trials. So we develop basically an experiment where we include people and we test our questions in the experiment. We take data, we analyze the data, and then we know if the treatment is working or not. So we do that in the first period in a phase 1 trials. And phase 1 trials usually have very few participants, sometimes are done in healthy people, not people with the disease. And those studies are mainly to analyze safety issues. We want to be able to detect if there are any safety issues with the drug that we're studying. So we're going to analyze what is the dose, how the drug is metabolized in the body, how it's eliminated, and particularly safety. Now, in a phase 2, we're going to keep collecting information on safety, but now we're going to add some measures on efficacy. So we want to know also if this drug is working or not in the measure that we want to measure. For example, if it is a drug for, let's say, cognition, I will use a metric to measure cognition, and then I will start getting data on the drug, if the drug is working on cognition, is improving cognition or not. And the other thing is that we're going to look for, identify a potential dose response. In a phase 3, the focus is on efficacy. And if in phase 2, there are hundreds of patients. In phase 3, there are usually thousands of participants. And here, we really want to identify if the drug is doing what I'm made to do. So if my focus is on cognition, I will have two groups. In all of these phases, you have a placebo group and a treatment group. And I'm going to measure, for example, cognition in those two groups, and I'm going to compare to see if my drug is improving cognition or not. If the drug, and also you take information about safety, but mainly the safety issues should have been resolved in phase 1 and phase 2, if there are major safety issues. Now, if there are no safety issues in phase 3, because we have more people, and that also help us to identify potential issues, and the drug is shows that it's efficacious, then that information is analyzed by regulatory agencies to grant approval or not. And after that, what we do is what is called phase 4 or post-marketing studies, when we analyze data on safety, particularly when there are millions of people taking the drug. So that information is also important, because sometimes after the approval of a drug, there can appear new adverse events that weren't detected in the previous phases. So what we did in 2022 was to start an analysis on what was the drugs that were being studied in clinical trials in Lewy body dementia from 2000 to 2022. And for that, we analyzed 412 records, and we identified 40 trials in those 22 years that were investigated 25 agents. That study was published in 2023, and we also analyzed the evolution of those clinical trials. So what we found was that there was an increased interest in trying to find treatments for people with Lewy body dementia. More than 50% of the trials were active in the last 10 years. Now, we also find that there were very few trials in phase 1. Phase 1 is important because we want to be able to have enough agents that will move along the rest of the phases. So we need to work on that. Sorry? I'm free. Is it working now?
Yes.
Okay, perfect. We also found that most of the agents that were being studied were repurposed agents. So that means there were treatments that have been approved for other diseases, and we were studying those treatments in Lewy body dementia. Most of the trials were focusing on mild to moderate dementia, which is okay, but probably we want to move to earlier stages of the disease. We also found that most of the outcome measures, or the way that we were measuring if the trials were working or not, we were using scales that have been developed for other diseases, and some of them need to be validated in Lewy body dementia. Very few trials use biomarkers as an inclusion criteria. Most of the biomarkers were in the exploratory phase. And we also found that there was a lack of diversity. And that is an issue, because we need to be able to test these treatments in all the population that is going to take them, to know if it's going to be efficacious or not. So we concluded that there were challenges and opportunities. One of them was to be able to detect patients early. The other one was to develop disease-specific outcome measures, and then also improve our ability for global representation and diverse populations in this clinical trial. At the end, we concluded that there is an urgent need for more trials. So now in 2025, what we are doing is we are updating that search. And what we have found from 2022 to January 1st, 2025, is that there are actually right now 10 active trials that there have been 13 recent completed trials and 8 other, meaning that they have been withdrawn or terminated. So there are other status of these trials. Now, I don't want for you to focus because this is super technical about what are the different drugs and what are the phases. But what I want to show you here, so in the inner circle, we have phase 3 trials. We said that those are the ones where the treatments are closer to the users. Then we have phase 2 trials in the middle and phase 1. Again, we see that there are some white spots, and that means that there are no trials in that area. So for example, we have not been able to find clinical trials for cognitive enhancers or for cognition in phase 1. Or we were not able to find also phase 1 trials for neuropsychiatric symptoms and behavioral symptoms in the Lewy body dementia or sleep disturbances. And there are no trials in phase 3 for sleep and disturbances. So there is a lot of opportunities here for improvement because it is annoying to do comparisons. But if we compare that pipeline with the Alzheimer's disease pipeline, we can acknowledge that there is a lot of work to do. So in Alzheimer's disease, there were last year, in 2024, 164 trials and in PD 136 trials. So how can we move along? What is the take-home message here? It seems like there is a lot of opportunities for us to do more clinical trials in Lewy body dementia and to move to early detection, because we want to move from a standard medicine where we identify a patient and give one particular pill to do a more personalized medicine. That will be a combination of the first part when we have biomarkers. So we combine this biological definition and the identification of people with biomarkers. We will be able to give them the right treatment at the right dose at the right time. So with all of this information on research, you might be wondering, so how can I participate in research, right? It's very important for us to understand the importance of people participating in research. So in Stanford, I work with Professor Kathleen Poston, and she has several studies. I listed here some of them. One of them is Lewy Bodies Spark, and the other one is the blood biomarker study. If you want more information, you can check the website. Also, Dr. Jory Fisher sent me this information about this beautiful study that she's doing that is called Persevere. This is an entirely virtual study, so fairly easy to do. It's for caregivers. And what she wants to do is to be able to identify information about the caregivers on a national level. So if you want more information, you can scan this QR code. Also, here you have the email. More things. The Lewy Body Dementia Association on the website has resources if you want to know more about the research centers of excellence, if you want to participate on trials, or what is the type of research that is being funded. So a lot of information there. If you want to participate and be active, just please go and check these websites. I want to finish by saying that it seems like there is a lot of things to do, but I think that we have come a long way and we're closer to the finish line. And emphasize the science is a team sport, and patients and the caregivers are in the center of our actions, and we need to work together as a team to be able to answer these research questions and to have better treatments for our patients. Finally, I want to thank my collaborators and my colleagues from the Boston Lab, and I'll be happy to answer your questions.
Okay, hold on, let's see. Okay. We have one question. Can you hear me? Carla?
Yes, yes.
We have a question from David. Could Dr. Abdelnour comment on the SIN1 skin biopsy test for alpha synuclein?
Yes, thank you very much. There is a lot of information on different biomarkers. I didn't get the chance to put them all because it's a lot. But yeah, so there is another biomarker that detects alpha synuclein in the skin. And it seems like it's very good at differentiating people with Parkinson's disease. Now, this biomarker, it seems so when you develop a biomarker, one of the things that you have to decide is what is your gold standard. So if you have a new test, what are you going to compare it to? So if you compare it to the clinical diagnosis, it seems like in this case, it's very good at detecting the clinical diagnosis. And that will be sometimes preferable for some people instead of the cerebrospinal fluid. But what probably needs to be done is to compare to neuropathologies because that's the gold standard for the diagnosis of neurodegenerative diseases. So as I mentioned, these diseases overlap a lot in the symptoms. So the definitive diagnosis is to have the brain tissue and look at the brain tissue under the microscope. So it would be great if in the future there is a validation of this biomarker against neuropathology.
Okay, thank you. Ryan asked, thank you for your presentation. Can you repeat what percentage of patients have the mixed Alzheimer's disease and LBD? And I'd be curious from a personal perspective because we always thought my father was a pure case of LBD.
Yes, so the percentages differ depending on the source. So if we look at studies that have analyzed brain tissue, so the tissue under the microscope, the percentage of cases that have Lewy body dementia and Alzheimer's disease is approximately 80% for amyloid, concomitant pathology, mixed pathology, and 50% for tau pathology with alpha synuclein. So the combination would be alpha synuclein with amyloid or alpha synuclein with tau. And in neuropathological studies, it's approximately 80% for amyloid, 50% for tau. Now, in biomarker studies, those percentages are lower, and is for amyloid approximately 50%, and for tau, it seems like it's around 30% or less. We don't know why we have this difference. We don't know if it is because when we do neuropathological studies, those are end-stage cases that are closer to death, so maybe they have more pathology. The other reason could be, it's okay. The other reason could be because the biomarkers are less sensitive to the burden of pathology that is present in the brain.
Okay, great. I'm having technical difficulties, so you can't see me, which is no great loss. But Lori did ask, hello, how do we find out more about the Stanford clinical studies?
Yes, so let me copy the information here. I'm going to put, because we have a website, the Boston Lab, where we have all the details, and each study have a contact person. So I'll put this in the chat. And this is the link to the website, the Boston Lab website. And there you have the details of this, the name of the study, the details of the study, and the contact person.
That's fantastic. Also, if someone is in Spain and interested in an LBD trial, how do they find out about that?
Yeah, so one option is to look at the website. So in Spain, there is a website that registers all the clinical trials. Let me remember the name. It's the AMEPS, is the agency in Spain, and that has a registry of the clinical trials. I check if I remember correctly, because I did that graph that I showed, I'm working on that paper right now. There is one clinical trial for PD with mild cognitive impairment right now active in Spain. I don't recall if there are specific trials for Lewy dementia, but maybe it's missing because I haven't got the chance to analyze those details. But if you want more information, you can go to the IMFs. I'll also write that on the IMFs. And that website has information on all the clinical trials for all diseases in Spain.
Now, Dorothy has shared a personal story, which is very interesting. It's about her husband. A few doctors think that he might have LBD. He had hallucinations and cognitive loss at first. They hired an exercise caregiver right away, and with the drug Nuplade and 6 months of working with the caregiver 3 times a week for 2 to 3 hours each time, he no longer has the hallucinations and his cognitive abilities have become nearly normal. Can you comment on that, Carla?
I don't know that drug, so I will have to take a look at the name of the drug, because I'm not familiar with it. Now, from the name, I don't recall reading it when I did the search on the drugs in Lewy body dementia. So I
So Carla, she
will
probably
be,
means
yes.
she probably means
Nuplacid.
Nuplazid.
Oh.
Yeah.
Oh, okay. Oh, okay. So Nuplacid, yeah. So that's a drug for hallucinations. So it explains why the hallucinations have improved. And people with Lewy body dementia fluctuate. So, well, yeah, it's possible that they're seeing, if the hallucinations were also causing a lot of impairment in behavior, in activities of daily livings, and now the hallucinations have improved. So maybe that's why they're seeing an improvement.
So from a personal perspective, Nuplacid did not exist when my father was having hallucinations. So is this a relatively new drug?
Yes, it's new.
Okay.
Yeah, it's very new.
So it was really fascinating to hear about the biomarkers, and that's a huge step forward. Dana asks, is AI currently involved, or is there a plan to use AI in the future for plasma biomarker studies and development or any other LBD research?
Yes, that's a great question. There is a lot of interest in using AI for analyzing this data, because biological data is very complex. So one of the methods that we're using right now is AI. Actually, in Stanford, I know that I'm not sure for particular this disease, but for other diseases, they're also trying to use large language models, like the models that are used for ChatGPT for analyzing complex data in medicine research, in medical research. So, yeah.
So this is a timely question from Ryan. How do you expect US NIH funding cuts to impact LBD and other neurodegenerative disease research trials?
Unfortunately, yes. Anything that impact our ability to do this type of research will have a negative impact on the patients and the caregivers, because there is an ecosystem where public universities or public sector receive funding from the NIH, for the federal government, and that is used for a lot of different studies. And then that information helps the private sector to invest in some of the things that they might think that would be profitable. And that will impact that ecosystem. That will impact different type of research that has to do with diagnostics, with the caregivers, how to take care of this type of patients, because it's not only about if this is profitable or not from the drugs perspective, right? It's also about how we improve the quality of life of these patients. So unfortunately, I will say yes. I think it's important for people to advocate for research and understand that this information is in science benefits all of us. All of us will benefit from it.
Yeah, I think it's also impacting on private universities and their research as well.
Yeah, yeah, because they get also grants from the NIH, and it's not only public. Yeah, it affects all the research academic centers that receive grants. Is if there is less investment in research, it will have a negative impact in the short and medium term, unfortunately. Yeah.
So as we wind down this session, Lori asked, you mentioned there is so much crossover with all the other diseases. Do you partner with the researchers performing Alzheimer's trials? There are so many studies when I visit the Alzheimer's sites.
Yes, yeah, we work with them. So in Stanford, we work very closely with the memory unit. I work with Dr. Poston in the movement disorder unit, but we work very closely with the memory unit and Dr. Sharon Shah, who is the division chief of the memory unit, is actually the PI on some of the clinical trials for dementia with Lewy Bodies. So we work very closely, and me personally, this clinical trial analysis pipeline, I'm doing that with the Professor Jeffrey Cummins, and he is the one that does the pipeline in Alzheimer's disease. So we're all in conversations and working together to understand better this overlap. Yeah.
Well, I would say that we had at least 60 participants on today's webinar, which is fantastic. And then those 60 people will share information, I'm sure, with friends and family. So exponentially, we could have reached out to more than 100 people today. So Carla, thank you so very much. For everyone who is participating, we will do a follow-up and include information from today's webinar. So if you took notes or not, you will still get a comprehensive summary of what was presented today. So Carla, any parting words from España?
Well, thank you very much for the opportunity, and I hope that this information is helpful, and I hope now that you feel more engaged and want to participate in research and be part of the scientific community.
Absolutely. And the more we can reach out to our representatives in Congress about research, the better, because only we can make that difference. Thank you so much for everyone's participation. The questions were excellent. Carla, thank you for your research and good luck with your continuing work.
Thank you. Thank you very much.
Bye-bye.
Bye.